Hello, so how do we use our experimental design so as to minimise the effects of the problems we've just been through, the problems of confounding variation? What we're going to do here is we're going to go through all the different ways of using our experimental design to do that, and we're going to look at examples of experiments that have done that. So the first thing we're going to look at is including the appropriate control. And not all ecological experiments need a control. And there are two kinds of control. There's a negative control, and you use a negative control when you are comparing the current situation, the unmanipulated situation, to a new situation where you've performed a manipulation. And the negative control is a group which is identical to your treatment group, but without the specific variable whose effect you want to test. So you have to perform absolutely everything that you would have done with the treatment group, except you stop short of performing the actual manipulation. So, for example, a sham operation, something like, for example, if your operation involved installing a piece of equipment like a pacemaker, you'd have to, as a control group, you would have to do everything, including going into anaesthesia, including opening up the patient, including identifying the place where you would install the pacemaker, and then you would just stop short of actually installing the pacemaker. You wouldn't install it, and then you would close the patient back up again, and that would be a control group. And that would allow you to see the effect specifically of installing the pacemaker and not the effect of doing all of the other bits of the operation. You can be sure that the effect of your experiment, the results of your experiment reflect the effect of the pacemaker itself and not of performing all of the initial bits of the operation. Similarly, a placebo trial. If your drug administration involves giving a pill, then a placebo obviously has to be a pill which just is not biologically active in the way that you want to measure. So that's a negative control. It's something which is identical to the treatment group without the specific variable whose effect you want to test. But you can also have a positive control, and a positive control is an extra treatment group which demonstrates that your test can produce positive results. So it's something that shows that you are able to detect the effect that you're interested in, something where you're certain that this treatment group is going to produce a positive result, and you include it so that you know you've kind of artificially created a positive outcome, so that when you look at your results from the treatments that you actually want to test, you have a positive outcome against which to compare it. So, for example, if you're looking for DNA in let's say a museum specimen, then and you want to be sure that it's possible for your procedure to detect DNA, you know that it's present only in trace amounts, and it could be that it's undetectable. But if you just have a bunch of completely negative results, then it could have been that your procedure had gone wrong. So you would include a positive control in order to show that your experimental procedure is okay. And your positive control shows up positive, and that means that if the rest of them show up negative, that you didn't do something wrong in your experiment. Another one is, for example, if you want to test whether a species responds to alarm calls from another species, you would use playback of the species' own alarm call to make sure that your playback procedure can actually elicit a response. So if you see your species responding to an alarm call, to its own alarm call, but then it's not responding to alarm calls of other species, then you know you've had that positive control, and you know that it's not something wrong with your speaker. Like, they can actually hear it and they can respond to it. They're not just kind of failing to respond to the speaker. Okay, so that is control. You only need a control where you're in one of those two situations, and you need to be able to compare your test results with either a negative control or a positive control. So now we're going to deal with pairing and blocking and randomization. And these are ways of allocating your treatment groups so as to minimize the effect of either common confounds that you know are going to be confounds, or unknown confounds. You know that your treatment groups are likely to, that your individuals' replicates are going to share things, but you're not quite sure what it is that they share. So when you know, the best way to divide subjects among treatments is either in a completely randomized design, when you want to just generally reduce the effects of confounds. That's a generally good idea. But then when there is a specific thing that you know is going to be a problem for your experiment specifically, you can use a paired or blocked design when you want to minimize the effect of a specific confounding variable. So thinking about a randomized design, let's consider the experiment of Anderson, 1982. So what Anderson did was he was looking at widow birds. He wanted to ask whether the tail length of widow birds had an effect upon mate choice. So what he did was he first chose some males for the experiment, and they all had like a sample of tail lengths, which were chosen completely randomly. So he randomly allocated males to the different treatment groups, and then he artificially shortened some of their tails and artificially lengthened others. And in a control group, he left one control group unmanipulated, thinking about this control group that we've just been talking about. But then the second control group, he performed the operation of cutting the tail, and then he stuck the tail back on to regenerate a tail of the same length. So he neither shortened nor lengthened the tail of that control group. And then when he looked at the results, he found that males with elongated tails had more nests initiated in the period when they had elongated tails than they had before. So on the left graph you can see the nests that they had at the start of the experiment, and on the right you can see the number of nests that were started with them, that were initiated with those males after the experiment. So you can see that there's a big difference, and the males with experimentally elongated tails had a lot more nests, got a lot more nests after the experiment than they had done before. The key point is that he had randomly allocated the males to those treatment groups, so he knew that he was he was deliberately randomizing any effects that the males might have shared among males that were allocated to a particular treatment group. And this kind of design, more formally, is known as a randomized controlled trial. So another example of a randomized controlled trial in ecology is Cowie et al., who were looking at the ecological effects of different management practices in reed beds. So they could either cut, looking at the effects of cutting the reeds and looking at the effects of burning the reeds upon the diversity of plants and flowers within the reed beds, and on the breakdown of leaf litter within the reed beds. And so what they did was they had a big plot. They divided it into squares, and each square they randomly allocated to one of the three treatments. And it's that random process of allocation that means that they're not, the different squares within the grids could very easily share different ecological characteristics. Like, for example, there might be a gradient of ecological conditions like temperature or light intensity, or one side of this grid might be more closer to the trees and closer to other kind of ecologically important factors. So the important thing here is that Cowie et al. Randomized the treatment group that was allocated to each of these blocks, so that no gradient from left to right or top to bottom could potentially drive differences in systematic differences in the results. And they found that burning and cutting positively affected the flower diversity but had no effect on the leaf litter breakdown. And that because of that randomized design, they can assign those differences to the practices of burning and cutting and not to any confounding variables. So that's randomizing your design. So pairing is something a bit different from randomizing. Pairing is where you choose experimental subjects that you know will share lots and lots of different things, and within a pair, you choose pairs of subjects that you know will share different things, lots and lots of unmeasured variables, and then you allocate to each of the pair different treatments. And you do that for as many pairs as you can possibly do. So here is a burying beetle, and burying beetles have parental care by both males and females. They have a delightful system where they bury a dead mouse and then they lay eggs on the dead mouse and chew it up into meat, and then they feed the meat to the developing offspring. But what Muller et al. Wanted to know was whether males actually help, because it's possible for a female to raise offspring on her own in this species. So they wanted to know what the effect of males was. And what they did was they chose two females from the same family, so sharing genetics, and they allocated those two females to different treatments. The first treatment, they mated the female with a male and allowed them to bury a mouse together, but then they removed the male. And the second treatment, they allowed both male and female to bury the mouse together and then to raise the offspring. So you have two groups there, but the treatment is paired because the subjects are arranged in family groups. So one member of each pair, both members of the pair come from the same family, but one member gets one treatment and one member gets another treatment. And so what they used that experiment to find that what you're looking at along the x-axis is the difference in offspring between the paired female and the singleton female. This is a histogram of what that difference looks like. And you can see here that there statistically was no difference between paired females and singleton females. So statistically speaking, the male had no effect in the numbers of offspring that the female laid. Now, there's subsequently been lots and lots and lots of other work on burying beetles. So there's very, very early work on burying beetles, and there's been loads of stuff subsequently. But it's a nice example of a paired design where they use two related females, each of which receives different treatments. It's a bit like the twin studies that you sometimes see, where the twins that share absolutely everything in their genetics, but just differ in one thing that the researchers want to look at. And that's why twins are so valuable for studies of differences. And then you have blocking. So blocking is a bit like pairing, but it's where you identify a potential confound like time or age or body size, something which could, if you didn't monitor it, could potentially drive your experimental results. And then you identify groups that share particular common values of let's say they're age cohorts or groups that are all similar in their body size. They don't have to be identical, they have to just be similar. And then within those groups, you allocate your treatment groups. So that would be the yellow treatment and the black treatment in this case. And then you make sure that you allocate your treatment groups evenly within each cohort, within each block. And then, as long as you've made sure that you do that, then at the end of the experiment, you can statistically combine all of the blocks, and you can make sure you can then be sure that the effect of your confounding variable has been accounted for, because you have looked across the entire range. You've made sure that your treatment groups have been allocated across the entire range of the confounding variable. So here's a good example. It's not really very important, the details of this, but Frank et al. Wanted to look at the effect of density of beetles upon the number of cutworms they ate. But the important thing here is that they, and whether they were given different kinds of food, cutworms and no food, cutworms and pupae, or cutworms and seeds. The important thing is that they blocked this by time. So they did the experiment at several points in the life cycle of the beetle, and they made sure that each of these treatment groups, all 6 treatment groups, were allocated evenly across all of the experiments at different time periods. And so they don't even need to talk about the effect of time, and it's not even shown in the key graph that they give you. But the key thing is that those treatment groups, all 6 treatment groups, were represented at all time points that Frank et al. Did their experiment. So at the end of the experiment, they could combine all of the time periods together, and they knew that that potentially confounding factor was accounted for. It wouldn't have been if they had done the no food experiment, if they'd done the black bars early and the white bars late, then any differences between the groups might have been accounted for by time instead of by the different treatments. But as it was, they blocked by time, and that's kind of statistically accounted for the effect of time, and they don't then have to talk about that. So each bar combines the results from all blocks. So the final thing I want to talk about is pseudo-replication, and that is about knowing, reducing pseudo-replication is about knowing the true unit of analysis, knowing the level of replication that's appropriate for the experiment, and ensuring that you have enough replication at that level and not at not at a lower level. So the key thing is that replicates are meant to be independent and randomly chosen, and if that's the case, then any random variation that occurs between individuals cancels each other out. But if replicates are not independent random samples, then that variation might not cancel each other out. So you might get related, some of your replicates might be related to each other in ways which affect your result and which doesn't cancel each other out. So for example, if you want to ask are athletes taller than non-athletes, one obvious way to do, an obviously pseudo-replicated study would be if I measured the same person 20 times and they call that a sample of athletes. That's obviously not an appropriate thing to do. Slightly less obvious is if I recruit my sample of athletes all from one particular sports club in the same area. And so it could be that it might not be because they're athletes, it might be because they are from that particular region, or it could be that particular sports club uses some technique or is has something in the food, or there's something about that particular environment that has affected the height of the athletes coming from that sports club. So that's another source of pseudo-replication. Or something really subtle. For example, let's say I promote my study. I'm trying to do everything right, and I'm trying to do things as particularly rigorously. But then I publicise my study over Twitter, and one local basketball team. I'm trying to recruit from lots and lots of different sports to look at athletes. But then there's a particularly active Twitter community among a local basketball team, and then they all sign up for the study and they end up overrepresented in my study. And so basketball players end up overrepresented in my study, and that inflates my idea of the height of athletes generally. So that's another kind of really kind of subtle form of pseudo-replication. That's, if you weren't aware of it and you didn't think about it, it might end up driving differences in your results which are not actually down to the effect that you want to measure. So pseudo-replication can be really, really subtle. So the way to get around it is to think about and know what the unit of analysis is that you're both that you're trying to measure, but also that you've actually got given your experimental design. And you need to make sure that that level receives adequate replication. And you want to make sure that the replicated units are independent. So like if it turns out that this is the level of replication, you need to make sure that you've got enough units to provide you with enough statistical power to claim that your result is real and not just and so that you can estimate the mean value for each treatment group with a degree of confidence that allows you to answer your question. So I've just taken some examples from papers. So these are just cut and pasted straight from the papers. To minimise pseudo-replication, we scored just one randomly chosen egg clutch out of any consecutive egg clutches laid in adjacent pools within the same site. To minimise pseudo-replication for each repeat female, we used data from only 1 year chosen at random. At least 1 hour was left between watches on the same individual to minimise pseudo-replication. Yeah, so pseudo-replication can even come from, you can have temporal pseudo-replication if you watch something and then you take another measurement too soon afterwards, then those two measurements can be pseudo-replicated because the individual hasn't had enough time to change its behaviour enough, like to provide you with an independent reading.